Thanks for joining me today. I am Michelle Carlson. I'm a product manager at Active Motif. And today I'm going to talk about the KRAS In-Well Lysis ELISA kit. This is a new product that was released in 2023 by Active Motif. And it's been designed for high throughput quantification of active KRAS in cell lysates. This was developed to address a particular need that we saw in the market. Before I go into detail on the kit, I just want to briefly talk a little bit about the background of KRAS. First of all, RAS is a frequently mutated oncogene that you find in 20 to 30% of cancers. The gain of function mutations occur at codons 12, 13, and 61. And these result in enhanced GTP binding or impairment of GAP, which are GTPase activating proteins. So virtually all mutations, 98%, are located at these codons. As a result of this, you see downstream increase in signaling, which drives tumor transformation and progression. Although all these RAS mutants are activating, they're not all equal in their oncogenic potential. Both the mutation frequency and patient survival rates vary with the different isoforms. So in particular, KRAS is the most frequently mutated isoform, and this accounts for 80% of all RAS-driven cancers. To make matters worse, KRAS mutations are especially high in some of the deadliest of cancers, like pancreatic, colorectal, and non-small cell lung cancers. As a result of this, there's a need for assays which rapidly quantify KRAS activity to determine the effect of potentially therapeutic compounds. People are really looking at isoform-specific compounds, and in particular KRAS, because it's so highly mutated. So we set out with the goal to create an assay for fast, sensitive, and highly specific quantification of active KRAS in ELISA format. And you can see here what we started with was to find a highly specific KRAS antibody, and this was achieved by screening recombinant antibodies cloned from antigen-positive B cells and isolated from rabbits immunized with a KRAS-specific peptide. In this slide, you can see data for the clone that was ultimately determined to be best for our KRAS In-Well Lysis ELISA assay. On the left-hand side, you can see results from a Luminex immunoassay. So native KRAS, NRAS, and HRAS proteins were separately coupled to optically distinguishable Luminex magnetic microsphere beads. And then these beads are mixed together with a serial dilution of the recombinant KRAS antibody and agitated. The beads can then be captured with a plate magnet so that any beads associated with a KRAS antibody can be detected using a pigment conjugated anti-rabbit secondary antibody and then read out on the Luminex instrument. So you can see in the figure that the median fluorescence intensity is reported for this titration of antibody. And you can see even at 2,000 nanograms per mil, the KRAS signal is huge. It's 25 times higher than what you see for NRAS or HRAS. And then on the right, we also did a Western blot And here you see 100 nanograms of either HRAS, NRAS, or KRAS recombinant proteins were denatured, and then they're run on SDS page gel in parallel, and then transferred to a nitrocellulose membrane. And then you can see this Western is split into two panels. The panel on the left is probed with 1 microgram per mil of a pan-RAS antibody. Panel on the right probed with our recombinant KRAS antibody. And you can see that the KRAS recombinant antibody is highly selective for KRAS on the right, and on the left, with pan-RAS antibody, you see nearly equal signals from HRAS, KRAS, and NRAS. So we determined that we found a KRAS antibody that's highly specific, and then we moved forward with this to see if we could find an automation compatible workflow. And this is an extension of a kit that we had already, the RAS GTPase KRAS ELISA kit. And we use that as a basis to see if we could eliminate some steps and make it a little bit more high throughput friendly. And you can see here the protocol that we created uses the RAS binding domain of human RAF. It's coded onto the assay plate, onto the ELISA plate. And then separately, cells in a 96-well culture plate are lysed by adding a 5x lysis binding buffer added directly to the cell culture media that's still in the plate, incubated for 15 minutes. And then at that point, the lysate can be transferred to the assay plate with the bound RAF RBD protein. And then that's incubated for an hour. The lysate is removed, plate is washed, the KRAS specific antibodies added, and then more wash steps are added before the secondary, and then a chemiluminescent substrate is added so that the relative light units can be measured on a plate reader. So you have the option in the fastest workflow, you add the 5x lysis binding buffer directly to the culture wells containing the media, but there's other options as well. So it's a flexible workflow. You can also, if you'd like, remove the media and then add a PBS wash before adding your lysis buffer. We did some comparison data to see what the effect of this was. And you can see on the right-hand side is this comparison data. In this experiment, ASPC1 cells were seeded at 80,000 cells per well, cultured for 2 days, and then treated with either none or 33 millimolar of MRTX 1133. That's an inhibitor of KRAS G12D. So they were treated for 3 hours, and then immediately for the purple bars, the media was removed, plates were washed with PBS prior to performing the in-well lysis. And then in tan, you can see the lysis buffer was added directly to the media in the wells with no wash. So both methods work, but you might see an improvement in the assay window if you do include the wash. So it kind of depends on what your requirements are. If you want to be faster, or if you require a little bit more signal depending on your cell line. In addition, you can do you can quantify the protein. Some people want to make whole cell extract and then quantify the protein before adding it to the ELISA. So you can do this with cells or tissue. Here I'm showing, the workflow for cells. And you can, with this, you could create a lot of cell lysate and freeze it and use it a number of times. So with this protocol, you remove the media, wash with PBS, add your lysis buffer, transfer your cells to a microcentrifuge tube, incubate on ice, pellet the debris, and then transfer the supernatant. And then once you have that, you can quantify the protein using either a BSA standard or Bradford-based assay. So here's some data using the In-Well Lysis protocol. This is the no-wash protocol where you just add your lysis buffer directly to the culture plate. It's a histogram showing the relative light units in which the ASPC1 cells were seeded at an 8.2-fold dilution series. So we started with 640,000 cells, and that was diluted down, cultured for 2 days, and then the In-Well Lysis assay was performed. At that point, so the relative light units were read on a plate reader, and then you can see clearly that the active RAS signal increases as the seeding density increases. This same assay can be used to do an IC50 determination. So this is exactly that same protocol, in-well, no wash. And this was used, again, this was done with ASPC1 cells. They were seeded at 320,000 per well, cultured for 2 days, and then they were treated with a broad dilution series of MRTX 1133. Then the In-Well Lysis protocol was done, and you can see in this graph, this is background subtracted. You can see this was done with four samples. So this was replicated 4 times. And then an IC50 analysis was done using GraphPad at Prism. Just to remind you, MRTX 1133, it's a KRAS G12D specific inhibitor. And this cell line expresses that particular mutation, that particular KRAS mutation. And using this method, we determined an IC50 of about 9.1 nanomolar. And this is in really nice agreement with data that's been reported that's out there for this inhibitor. In addition, you can, as I mentioned before, you can just use whole cell extract and quantify it and then add it to the assay plate. So here, this is what was done. This is, again, the ASPC1 cell lysate. So they were lysed, then they were quantified using the Pierce BCA protein assay kit. Then they were diluted in 1x lysis binding buffer, and 50 microliters was added to each well. So you can see here really strong signal from the active KRAS as you increase the amount of lysate that's added. And in addition, you can also see that the assay window is increasing. So the assay window is on top of the bar. So you can see it goes from 5 to 28. So if you need a higher assay window, yeah, you may need to increase the amount of lysate that you're adding. This is some data that was given to us by a customer where they used tumor samples. So these were 5 human tumor samples in mice, and some of these were treated with an inhibitor, and others were not. So here, the inhibitor is AMG 510, and that was given at 20 milligrams per kilogram. So the tissue samples were harvested and frozen, and they were able to, pulverize the tumor into a powder in the lysis buffer, and then they spun these down and transferred those lysates to the ELISA after quantifying them. And you can see the effect very nicely here of the inhibitor in the samples that were in the tumors that were treated. The ELISA is produced in two different forms. So there's a 1-plate kit where you get enough for 96 reactions. And then we also have a 5-plate kit. The 5-plate kit has a discount built into that. So, and also we are selling the 5X lysis binding buffer separately. So some customers, if you want to do some experiments with different types of samples and freeze your cell lysates in advance, you can purchase that separately if you'd like. So just to summarize the highlights, it's an HTTP friendly workflow. You can do the in-well lysis workflow, and it will be work very easily that way. We also provide extra reagents. I didn't mention that earlier, but we give you like a 20% excess in the reagents. So if you want to use an automated plate washer, you can do that. It's highly specific for KRAS, which is something that people have been asking us for. And then, you have different flexible options for the cell lysis. So thank you very much. If you have any other questions, you can go to our website and see we have more information online about the ELISA. You can also contact technical support, and they can help you with any questions that you might have. And I really appreciate you taking the time to tune in with us today. Thanks.